Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kame Biology. Today in this video lecture, we are going to talk about DNA structure or Watson and Crick model. Okay, in the previous video lecture, if you guys remember, we talked that nucleic acid and we also talked the central dogma. Okay, for understanding of DNA structure or whenever we want to understand about the Watson and Crick model, we need to understand the basics. Now, these are the basics that what are actually nucleic acid, what are the nucleotides, what are the nitrogenous bases, pentose, sugar, phosphoric acid, how the nucleotide exactly will look like, the structure of the nucleotide. We need to understand the structure of each nitrogenous bases and everything. So I already explained these things in a detail. So first of all, you watch this one video. So it is present on my channel, you can watch it, or don't worry, I will give the link in the description box, then you will check it. So today in this video lecture, we are going to talk about Watson and Crick model or DNA structure. So let me erase this one portion and let me start. Okay, so let me write DNA structure or Watson and Crick model and Crick model of DNA. Okay, before going to explain the structure of the DNA, that how exactly the DNA will be look like, let me tell you the historical view, or let me tell you the history of the DNA. Okay, DNA was first of all discovered by a scientist. His name was Friedrich Mischer, 1869. He was a German chemist and he discovered the DNA. How he exactly discovered the DNA? He take the human cell and he also take the sperm cell of the fish, and he isolate the white substances. Inside the nucleus, he isolated the white substances. These white substances, he called them nuclein. Okay, and after some time, he called these white substance nucleic acid. Now, why we called it nuclein? Because it was related to the nucleus. It was actually isolated from the nucleus, that's why. And why after that it was called nucleic acid? It was called nucleic acid because they have the acidic property. That's why he called the DNA that it is actually nucleic acid. And then another scientist, his name was so we have another scientist, his name was Zacharis. Okay, the term of the DNA was discovered by Zacharis. Okay, so they give the name DNA. So the term DNA was discovered by Zacharis. Okay, Zacharis. And the DNA structure, the whole complete structure of the DNA was actually discovered by Watson and Crick. They discovered the structure of the DNA. Okay, now how exact the DNA will be look like? Okay, now remember, in DNA we have nucleotides, nucleotides. Usually in DNA we have the nucleotides, and in case of DNA, each nucleotides are nitrogenous bases, and in nitrogenous bases the nucleotides are actually, in nitrogenous bases we have adenine, guanine, cytosine and thymine. These are the nitrogenous bases in case of DNA. Okay, and they have also the phosphoric acid, and they have also the pentose sugar. But they have pentose sugar, we call that deoxyribose sugar. They have pentose sugar, we call that deoxyribose sugar. We already talked that how nucleotide will be look like. Now in this case, I will draw that how deoxyribose sugar will be look like, and how this nucleotide will be look like in case of DNA. So if I draw the structure again here, so in this case we have again this pentose sugar. Here is the nitrogenous basis. So it may be adenine, guanine, cytosine and thymine. Okay, so here will be the nitrogenous basis. One nitrogenous basis, one pentose sugar will contain one nitrogenous basis. Now this is called deoxyribose sugar. Okay, as we talked that why we call it deoxyribose sugar, because at carbon number 2 we have no oxygen, so oxygen is removed, that's why we call it deoxyribose sugar. Now in DNA we have this one sugar. Okay, so whenever this is called the nucleotide of the DNA, understand, this is called the nucleotide of the DNA. So when one nucleotide of the DNA connected with another nucleotide of the DNA, and then it is connected with another, and then it is connected with another, and so on, and they make a long polymer chain, we call that structure of the DNA. This is how the DNA will look like. Okay, but as we are explaining the Watson and Crick model, you know, Watson and Crick explain the structure of DNA very well, and they explain a complete structure of the DNA. According to them, they talk that DNA is a double stranded. So how it will be look like? It will be look like in this form. Okay, so according to Watson and Crick, he talked about the DNA is a helical in shape. So it is the double helix. Helix means helix will be look like in this form. So they talk that DNA is a helical like shape, and DNA is made up of two strands. This is the first strand and this is the second strand. Okay, so they says that the one strand have 5' to 3' end, means this one strand have 5' end to 3' end, while the other strand have 3' end to 5' end. Fine. Now second points, they says that DNA is a double helix, and one thing more, they have double stranded, as you are watching, and the DNA is not, the two strands are not crossing to each other. Okay, now if it is the DNA, so it doesn't mean that they are crossing to each other. I mean DNA will be not crossing to each other like that. They are actually just like that. So DNA are not crossing to each other. DNA, the two strands of the DNA are not crossing to each other. It will be perpendicular, it will be parallel to each other. Okay, so if we see the DNA structure, so it is actually the parallel like in shape. This one, this is the parallel like. Okay, and we also say that the DNA is also anti-parallel. So they say that DNA is a parallel, and they also say that DNA is an anti-parallel. What does it mean? It means that DNA is a parallel because the strands are not crossing to each other. That's why they say that DNA is a parallel. They say that DNA is anti-parallel because his one strand have 5' end to 3' end, and his other strand have 3' end to 5' end. Okay, so both strands are opposite to each other. That's why they call that DNA is a anti-parallel to each other. Okay, next point they says that DNA is made up of nucleotide chains. Usually this one sugar molecules are the nucleotide in case of the DNA. So deoxyribonucleic acid, this one is the nucleotide. It is connected with another nucleotide, then it is connected with another, and they make a long chain. Okay, so according to them, according to Watson and Crick, they says the DNA is made up of nucleotide chains connected to each other and make a long polymers. So in this case, they have also purine as well as pyrimidine. So they have the nucleic acid, they have the nitrogenous bases, which we call that purine and pyrimidine. Purine, again, the purine is a double ring structure. And this pyrimidine is a single ring structure. So we call that dicyclic and we call that monocyclic. Okay, and they have nitrogen atom also, which is placed on carbon number 1 and carbon number 3. They have also nitrogen atom. So they have 4 nitrogen atom, which is placed on carbon number 1, carbon number 3, and carbon number 7 and 9. Okay, they have purine, we call that adenine and guanine. Fine. They have pyrimidine, we call that cytosine and thymine. Okay. Moreover, they says that according to this model, they says that adenine will always make a bond with thymine, and they make double bond, double hydrogen bond with thymine. And they says, according to them, according to this model, they says that guanine will make a bond with cytosine. So they make a triple hydrogen bond with the cytosine. Now, as we talk that when one nucleotide are connected with another nucleotide and then another nucleotide, then they make a long chain. But how exactly it will look like? Let me draw each nucleotide of the DNA, due to which it will more clarify the structure of the DNA. So as we talked, according to this model, we talked that DNA is made of nucleotide, which is the which we call that deoxyribose sugar. So how exact it will look like? It will look like in this form. So this is the first nucleotide. Okay, let me draw and here we have the hydrogen. This is the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and we have carbon number 5. Fine. Now at carbon number 1 we have the nitrogenous bases, as we talked. Carbon number 2 we have the hydroxyl, we have the hydrogen atom. Now at carbon number 3 we have a hydroxyl group. Fine. At carbon number 4 we have again hydrogen and again we have OH. And similarly at carbon number 5 we have the phosphoric acid. So CH2, they are connected with the phosphodiester bond, and here is the OH and OH, and one OH is removed from the phosphoric acid while the other OH is removed from this pentose sugar, and it is removed in the form of water molecule. So that's why the phosphodiester bond is made. Okay, this nitrogenous bases will be connected with a glycosidic bond, we already talked, and phosphate group will be connected with this sugar molecule we call that phosphodiester bond. Okay, now this is the first nucleotide. Now another nucleotide will be connected and make a phosphodiester bond at carbon number 3. This is the carbon number 3 position. So if the other nucleotide which is connected at the first nucleotide, it will be connected. Let me draw another nucleotide here. So here is again nitrogenous bases, H, H, OH, H, and again CH2 P OH. Okay, now again we have what? We have the let me draw. Now again we have the nucleotide. Now we have another nucleotide. Now this nucleotide will be connected with the first nucleotide. How? They will be connected and make a phosphodiester with the first nucleotide when the water molecule, when the OH are removed out. So whenever the OH are removed out, so they make a phosphodiester bond to each other. So then the first nucleotide will be connected to another nucleotide. That's how the nucleotides are connected to each other again and again, and they make a long polymer. Okay, so this is how the nucleotide is connected, and this bond is called a phosphodiester bond, as we talked here. Okay, let me once again tell you, this is the first nucleotide. They have phosphoric acid, nitrogenous bases, and this is hydrogen, due to which we can identify that this is the deoxyribose sugar. This is the first deoxyribose sugar, which we call the nucleotide of the DNA. It is connected with another nucleotide of the DNA, which we call the deoxyribose sugar. When add carbon number 3, okay, so at carbon number 3, there will be the bond formed, which we call that phosphodiester bond. So the second nucleotide will be connected with the carbon number 3, and they make a phosphodiester bond. So just like that. Then we have again a third nucleotide will be connected just like that fashion. Then fourth nucleotide, fifth, and so on, due to which a long chain is formed. Okay, so this is how they are connected. This area, as you are watching, it means this is the 5 prime, means the phosphate group will be connected at carbon number 5. That's why we call that 5 prime. This area is the carbon number 3, so we call that 3 prime. Okay, this one is the 3 prime because here is the carbon number 3. Now, as we talked that DNA are anti-parallel, so it means the next nucleotide will be opposite, means the next strand will be opposite to the first strand. So if we draw again each nucleotide, but here we will draw the opposite strand. So here we will draw the reverse, means just like in this one. Okay, so this is the phosphoric acid, and if we draw another strand just like that, if we draw another nucleotide just like that, so again we will draw an opposite form. Okay, now the same nucleotide, whenever they are connected to each other, so they will be connected with the carbon number 3 of this one nucleotide. So again we get the phosphodiester bond here by the removal of water molecule. So again we get the phosphodiester bond. So this is the phosphodiester bond. Now then again we have another nucleotide is connected. So one strand will be going in this direction, the other strand will be going in this direction. Okay, and if we talked about the nitrogenous basis, so let's say here we have the nitrogenous basis, we call that, if I write here the exact nitrogenous basis, so let's say here we will adenine. So definitely they will make a bond with thymine, as we talked, and they make a double bond with thymine. If I remove here the nitrogenous NB and I write the exact nitrogenous basis, so here will be cytosine, and they make a triple bond with the guanine. Okay, So this is the exact structure of the DNA, that how it will be look like. Fine. So in this case, if you are watching, so this is the carbon number 3, so we call that 3' area. This is carbon number 5, so we call that 5' area. Okay, so that's why we can say that its one strand is going to 5' to 3', while the other strand is going to 3' to 5'. That's why we call that it is anti-parallel to each other. You know, according to this model, they says the pentose sugar and they says the phosphate group are what? Pentose sugar and phosphate group means these two are what? These two are the backbone of the DNA. Actually, DNA is a skeleton, so we can say that according to them, pentose sugar and the phosphate group are the backbone, while these, you know, these adenine, thymine, cytosine, guanine, these nitrogenous bases, they say that these are the cross bridges. So according to them, the nitrogenous bases are actually the cross bridges. They also describe the diameter of the DNA. So they, you know, watch the DNA at that position. Let's say if this is the DNA, so they watch the DNA at that direction. So at that direction, they say the diameter of the DNA is 220 Augstron. Now if we write it in nanometers, so they says that diameter is 2.0 nanometer. That is the diameter. They also says that DNA contain minor groove and the major groove. Now this is the minor groove as you are watching. This is the minor groove, and as you are watching this one, the whole, this is the major groove. Okay, so according to them, they say that DNA have two groove, which we call it minor groove and major groove. They say that the length of the DNA according to per turn means whenever the DNA is turned, usually the DNA is turned in a clockwise direction. Clockwise means DNA will be turned in this form, clockwise direction. Okay, so when the DNA turn in a clockwise direction, they says that, so in one turn they take 34 Augstron length. In one turn they says that they take 34 Augstron. If we see, if we write it in a nanometer, so it may be 3.4 nanometer. Okay, according to them, the distance between this means the distance between the nitrogenous bases with another nitrogenous basis means the distance between them. They says that the distance is almost 3.4 Augstron, and if we write it in nanometer, so it may be 0.3 nanometer. So this is the distance between the nitrogenous bases. Okay, one thing more that I forget here, that whenever the DNA is perform one turn, when the DNA is performed, when the DNA is circulated and when they perform one turn, so in one turn they have 10 bases, means they have 10 bases. If we write here, that says the DNA is turned one time. So when the DNA turn in a clockwise direction one time, so they have 10 nucleotide. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so they have 10 they have 10 nitrogenous bases in one turn. 10 base pair. They have 10 base pair in one turn. Okay, 10 base pair. Similarly, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6. So that's all about the DNA structure and Watson and Crick model. I hope you get idea about that, how the DNA will be look like, how the DNA structure will be look like. Inshallah, in the next video lecture, we are going to see the RNA structure. Same RNA structure will be also the same, but the difference is just that RNA have ribose sugar. We'll be seeing the RNA structure in our next video lecture. So thank you so much for watching this video. If you like this video, so make sure to hit the like button, share this video and subscribe the channel. Thank you so much.